Welcome, curious minds, to the deep dive. Today we're embarking on, well, quite a crucial investigation, diving headfirst into a world of invisible threats, stuff that can lurk in our food.
That's right.
We're talking about mycotoxins, specifically Aflatoxin B1, let's call it AFB1, and sterigmatocystin, or ST.
Two really potent compounds.
Exactly. And they're widespread dangers in common foods like maize, peanuts, things many of us eat regularly. And what makes this even more urgent is climate change. It's shifting growing conditions, right? Creating new breeding grounds for these contaminants.
It's definitely amplifying the problem, making it a truly global issue now.
So our mission on this deep dive is to really dissect a brand new study. I mean, this is hot off the press, February 2025.
Very recent.
From Pinepa Thongarm and her team. And this research, it's set to challenge a really long-held assumption about these two mycotoxins, AFB1 and ST, because they look so similar, structurally speaking.
Right. On a molecular level, they share key features.
Okay, so let's unpack this. For years, scientists, well, they largely believed that AFB1 and ST, because they have this similar warhead structure, would basically act the same way inside our bodies.
That was the prevailing logic, yeah. Similar structures, similar function.
But this new study reveals something pretty startling. They actually leave distinctive mutational fingerprints on our DNA.
Completely different signatures.
With really profound and honestly surprising implications for our health. Imagine that two toxins looking almost like twins chemically, but telling totally different stories inside our cells.
It really is like a molecular detective story, uncovering hidden clues.
Absolutely. So before we really get into the nitty-gritty of the study, maybe we should just set the scene a bit. For listeners who aren't deep into toxicology, what exactly are AFB1 and ST? Why should we care about these specific ones?
Good idea. So fundamentally, AFB1 and ST are what we call secondary metabolites. Basically, they're byproducts made by certain Aspergillus molds.
The kind you find on crops sometimes.
Yeah. Exactly. Especially crops stored poorly, maybe in humid conditions. And honestly, this isn't an exaggeration. These things are a global threat. Food safety, public health. They impact both.
And the health risks?
They're serious. Even low-level exposure over time can lead to things like severe liver damage, certain cancers, especially liver cancer, hepatocellular carcinoma, and it can even suppress your immune system. They're quite insidious.
Yeah.
And what's fascinating and relevant to this study is that for a long time, the focus was just on their shared warhead. This chemical structure everyone thought was the main source of the trouble.
It made sense at the time, I suppose.
It did. It made perfect sense to assume similar outcomes. Think of it like this. They both start from a similar basic building block, a polyketide precursor. And they both have this crucial common part, the difuranocoumarin core. That's like the key that unlocks their danger.
Okay.
Now, our liver has these enzymes, cytochrome P450s. They're our body's detoxifiers. They try to break these toxins down.
But it backfires.
In a way, yes. It inadvertently converts these cores into highly reactive epoxide forms. It's like flipping a switch that makes them chemically aggressive. These epoxides, formed right at the end of the molecule, were what scientists thought made AFB1 and ST so similar in damaging DNA.
How do they damage it?
They form covalent DNA adducts. Basically, they stick strongly onto our DNA, our genetic code, especially onto the guanine bases in the ATCG alphabet.
Got it. So they stick on, and that causes problems.
Oh, precisely. That was the traditional understanding. Sticky epoxides, similar damage.
And this, I think, is where Thongarm's new research really starts to shake things up. They used a pretty innovative method, right? These gptC57BL6J mouse embryo fibroblasts, MEFs.
Yes, MEFs.
Yeah.
It's a sophisticated cell culture system.
Sounds complex.
It is. But as the researchers noted, it's actually much easier to use and more efficient than classical intact animal models. Yeah. It lets you do faster, more controlled experiments right there in the lab dish.
And crucially, they added something to mimic the body's processing, didn't they?
That's key. They added a microsomal activation system. This includes what's called an S9 fraction, basically a cocktail of liver enzymes, often from rat livers and NADPH, which provides the energy for those enzymes to work.
So it's like having a little bit of liver function right there in the dish.
Exactly. It allows you to see how the toxins behave before the body processes them and after. It mimics that metabolic activation step.
Okay, so methodology set. What was the first surprise they found? Something about just cell killing. Cytotoxicity.
Right, and this was a big one.
Yeah.
Without any metabolic activation, so just the raw toxin hitting the cells, ST, sterigmatocystin, showed enhanced dose-dependent baseline toxicity.
Meaning,
at any given dose, ST was simply more toxic to the cells than AFB1, straight out of the gate, before the liver enzymes even got involved.
wow, okay, that immediately throws a wrench in that they're the same idea, doesn't it?
Absolutely. That intrinsic cytotoxicity of ST was a real revelation. It points to ST doing something damaging beyond just sticking to DNA.
Like what?
Well, the study suggests ST is strongly linked to generating reactive oxygen species, ROS.
Oxidative stress, free radicals.
Exactly. Think of ROS as unstable molecules that can damage cell membranes, proteins, and DNA, just causing widespread chaos and dysfunction. That could explain why ST is more toxic on its own before activation.
And AFB1, what happened when they did activate both toxins with those enzymes?
Both became more toxic, as you'd expect, but AFB1's toxicity ramped up dramatically. At higher doses, it caused almost complete cell death. This really shows how much AFB1 relies on that enzymatic activation, that switch being flipped by the liver, to become truly dangerous in terms of cell killing.
Okay, so ST is more toxic initially, but AFB1 becomes much more lethal after activation. Interesting divergence already.
Definitely. That intrinsic toxicity difference is significant.
Right.
But then the picture changes again when you look specifically at mutagenicity, the ability to cause DNA mutations, not just kill cells.
Right, because cell death and mutation aren't always linked one-to-one.
Precisely. And this raises that key question. If ST is more intrinsically cytotoxic, why isn't it causing more mutations? Or is it?
What do they find?
Here's the flip. AFB1 showed a strong, very clear dose-dependent increase in mutation frequency. More AFB1, more mutations. Predictable.
And ST?
Only a modest increase in mutations. And crucially, no clear dose-dependent trend. It wasn't like more ST equals proportionally more mutations.
So AFB1 is the bigger mutagen, especially after activation.
Consistently, yes.
Yeah.
The study concluded AFB1 shows significantly higher mutagenic activity. It seems AFB1 gets converted more readily into that super reactive mutation-causing form. It's a more potent genetic code shifter.
Okay, so different cytotoxicity profiles, different mutagenicity profiles, but you said earlier they do some similar damage. Is there any common ground?
There is. Despite all these differences, both AFB1 and ST predominantly cause one specific type of mutation, a gcETA transversion.
Okay, remind us what that means in DNA terms.
Imagine DNA as a ladder. The rungs are base pairs. G always pairs with C, and A always pairs with T. A GCA transversion is when a GC pair rung gets fundamentally changed into a TA pair rung. It's a specific kind of swap in the genetic alphabet.
Got it. And how does that happen? What's the mechanism?
It goes back to those reactive epoxides sticking to the guanine G, bases the DNA adducts. When the cell tries to copy its DNA, that adduct messes things up. Replication machinery gets confused by the damaged G.
And makes a mistake.
Yes. Instead of putting the correct partner C opposite the damaged G, it often mistakenly inserts an A. This is a known phenomenon called the A rule. It's a common error when dealing with this type of damage.
I see.
Then in the next round of copying, that mistake gets locked in. The original GC pair is permanently replaced by a TA pair. The study also mentions specific AFB1 adducts called FAPY adducts, which are particularly nasty. They stick around longer and are very good at causing these mutations.
Okay, so they both cause this GC to TA switch, but this is where the fingerprints really diverge, right? Where they cause it.
Exactly. This is the core of the distinctive fingerprint finding. AFB1 leaves a very precise address on the DNA. It's almost like graffiti tagging specific spots.
What kind of spots?
Its mutations cluster heavily in specific sequences, primarily 5-URCGC30 and secondarily 5-URCGC3. The G in the middle is the target. It shows a clear preference for these guanine-rich neighborhoods in the DNA sequence.
Like it can only fit or react well in those specific contexts.
That's the thinking. AFB1 has a part of its structure, the cyclopentenone ring, that's a bit puckered, not perfectly flat. This shape is thought to allow it to interact more selectively, maybe slide into or bind preferentially to those CGC or CGG sequences before the chemical reaction happens.
A key fitting only certain locks.
A good analogy. And this isn't just theoretical. This exact pattern, this specificity, has been seen again and again in different mammalian systems. And crucially, it perfectly matches something called cosmic signature, SPS24.
Cosmic. That's the big database of cancer mutations.
Exactly. SPS24 is like a known mutational barcode strongly linked to human liver cancer, hepatocellular carcinoma. Finding that AFB1 consistently produces this SPS24 signature in controlled experiments is like finding the molecular smoking gun.
Wow. So it directly links AFB1 exposure to the specific mutation patterns seen in liver cancers.
It provides incredibly strong evidence for that link. It's a powerful biomarker, connecting environmental exposure directly to a cancer signature.
Okay, that's AFB1's very specific fingerprint. How does ST's compare? You said it was different.
Very different. If AFB1 uses a fine-tipped pen, ST uses a much broader brush.
Meaning?
While ST also causes those G to T mutations, they're spread out much more uniformly across different DNA sequence contexts. There's only a very modest preference for 5-VOST CG3 Win, nothing like AFB1's sharp hotspots.
So less specific targeting.
Right. And maybe even more interestingly, ST causes a wider variety of other mutations too, things like GCCG, ATGC, GCT substitutions. It's a much more diverse, scattered pattern of damage.
And does that pattern resemble anything known?
Yes, it looks a lot like mutational patterns caused by oxidative stress.
Ah, back to the ROS connection.
Exactly. It fits perfectly. Remember, ST generates those reactive oxygen species. That likely contributes to this broader, more diverse mutation spectrum, hitting different bases and causing different kinds of changes beyond just the direct adducts.
So AFB1 is mainly direct adducts at specific sites. ST is adducts plus oxidative damage, more spread out.
That seems to be the picture emerging. ST's structure, its planar xanthone ring system, is flat, unlike AFB1's puckered ring. This flatness might let it slide into DNA less specifically, hitting more places, and its mutation profile actually aligns with cosmic signatures like SPS18 linked to oxidative damage, and even SPS4 and SPS29.
Which are linked to?
Tobacco exposure, interestingly enough.
Tobacco, how does that connect?
Well, it turns out STs has actually been found as a fungal contaminant on dried tobacco leaves. So that surprising link might have a direct environmental explanation. It really highlights ST's dual mechanism, direct DNA binding and indirect damage via oxidative stress. It's fighting on two fronts.
Fascinating. So let's pull back a bit. We have these two related toxins, initially thought to be similar, but this study shows really different cytotoxicity, different mutagenicity, and these unique DNA fingerprints. What's the bigger picture here? What does this all mean for us?
The implications are pretty significant, really, across several areas.
Like public health?
Definitely. First, these distinct fingerprints could serve as early-onset biomarkers. Imagine you could test someone's blood or tissue and see not just if they were exposed to a mycotoxin, but which one, based on the mutation pattern.
That would be huge for early detection, wouldn't it? Catching exposure before cancer develops?
Potentially, yes. It could revolutionize how we monitor exposure and risk, allowing for much earlier intervention.
And what about regulations, food safety?
That's another critical area. AFB1 is already pretty strictly regulated in many parts of the world. The US FDA has limits. The EU has even stricter limits, reflecting its known danger.
Right.
But ST is often much less stringently regulated, if at all. And this study really forces us to ask, are we underestimating ST's health impact because its fingerprint is different, maybe more complex?
Given its intrinsic toxicity and that dual damage mechanism.
Exactly. We likely need a more nuanced approach to regulating both toxins, maybe even considering combined exposures. This means better detection methods for our food supply, better storage practices to prevent mold growth in the first place.
Especially in vulnerable regions.
Absolutely. This is a global food safety challenge, particularly in warmer, more humid climates. And climate change is only making it tougher. We need better agricultural practices and storage solutions worldwide.
And beyond public health, what about just understanding cancer itself?
These findings are incredibly valuable there too. AFB1 and ST, with their now clearly defined distinct mechanisms, become really useful models. They help us tease apart the complex interplay between direct DNA adducts, oxidative stress, and the specific mutations that drive cancer development.
So understanding these toxins helps us understand fundamental cancer processes.
Precisely. Knowing exactly how each toxin damages DNA helps us develop more targeted prevention strategies. Maybe specific antioxidants against ST's oxidative damage, or ways to enhance DNA repair for AFB1's adducts. It pushes us towards precision prevention.
It's really incredible how drilling down to the molecular level can have such broad implications. This deep dive has shown us how AFB1 and ST, looking so similar, tell such different stories on our DNA.
The power of high-resolution mutation analysis is really clear here, moving beyond assumptions to see the actual genomic fingerprints.
Absolutely. It gives us precise, Actionable knowledge. But okay, here's something for you, our listener, to ponder as we wrap up. AFB1 and ST are just two members of this big aflatoxin family, right?
That's right. There are others like M1, G1, Q1, P1, all with slightly different structures.
So the question is, what unique fingerprints might they be leaving on our DNA? What other hidden health risks might be lurking there, waiting to be uncovered? Perhaps that's a deep dive for you to explore next.
It's a crucial area for ongoing research.
Definitely. We encourage you to keep exploring the world of mycotoxins and environmental health. Understanding these invisible threats is truly the first step toward protecting ourselves.